about B cells. So we're transitioning from T cells to B cells. We'll talk a little bit about just a high-level overview. We don't have to go into as much detail as we went into when we talked about T cell development, but we will build on some of what we talked about with T cell activation when we talk about B cells here. So just to give an overview, again, for step 1 purposes, you don't really have to know quite as much detail as we did when we talked about the T cells kind of going through the thymus, positive selection, negative selection. Not the case really with B cells. Now you might have learned more in your immuno courses, really for B cells in general in terms of their development. The big thing that you have to know is, again, they originate from the bone marrow, just like a lot of the other hematopoietic stem cells that then go on to form lymphoid progenitor cells. Now the B cells, like the T cells, will also have precursors. The difference is, remember, the T cell precursors are going to get chemokines that essentially cause them to migrate to the thymus, whereas the B cells, these are going to stay in the bone marrow to mature. That's the key difference. Remember, B for bone marrow, B for B cells, T for thymus, T for T cell. Now, once these B cells mature, and there is a process that goes along with that, but it's not particularly high yield for step 1. Again, you might cover it in your immuno courses, but not particularly high yield for step 1. The big thing here is that once these B cells stay in the bone marrow and they mature, then they become naive mature B cells. Now remember, you be careful with your terminology here. Remember, naive means that this particular B cell has not come into contact with its antigen. Now here we're essentially looking at an antibody. And what's really interesting here is that the B cell receptor is going to actually be membrane-bound immunoglobulin. Okay, so in other words, you're going to have membrane-bound antibodies on the surface of the B cell. So you can kind of see that here. You can see these antibodies that are bound on the surface of the B cell. And these antibodies are essentially going to be your B cell receptor. Okay, so this is a little bit different than T cells. T cells didn't have antibodies bound on their surface. And again, we'll talk about antibodies in great detail here, but you can see that antibodies make up a variable region up here, and there's a constant region on the bottom. And then there's also a couple different mechanical components here that we can see. We can see that there's a light chain here, and then we can also see that there's a heavy chain down here. Okay, and like I said, we'll talk about all of this in great detail, but the point that I want to convey here is that the antigen binding site, where your antigen is going to bind, is going to be up here in this region. Okay, so the antigen binding site is going to be up here in that variable region, and that variable region is going to be generated through somatic recombination of the VDJ segments as these antibodies are formed. And again, we'll talk about all of that. We touched on it a little bit in the adaptive immunity section, but we'll talk about that in great detail in the antibody video. Now, the other thing that I want you to kind of keep in mind here with B cells is there's usually going to be specific markers that call you to this being a B cell. In this case, CD19, CD20, CD21. Okay, so these are all representative of your B cell. And so when you see these markers, I want you to start to think about B cells. And you might remember, if you watched the video on T cells, that there are specific markers for T cells. And the specific marker for T cells is going to be CD3. Sometimes we'll take, a sample from a patient and we'll run it through flow cytometry just to see how many B cells, how many T cells that patient has. And, just for example here, let's say that I put on this side, let's do CD3. And on this side, let's just say I did CD19, for example. Okay, I'm just using one of these B cell markers. It could be CD20, CD21, right? But the point is that, you have to recognize, okay, this is CD19. So when I see CD19, what are they trying to tell me? They're trying to tell me that this is a B cell. Okay, so when you see CD19, you're thinking B cell. When you see CD3, you should be thinking T cell. And so you can get these graphs where you'll see like a bunch of dots, for example, here, and you might see a bunch of dots over here. And essentially what this is representing when you see these diagrams is it's showing you that, hey, this person has, some amount of B cells and T cells that were identified when flow cytometry was conducted. The point that I just want to hit home here, and we're going to go into this in a lot more detail when we talk about how to read these graphs and that type of thing. But the point here being, if I gave you this and I had a whole bunch of density here at the CD3 line, but I had maybe 1 or 2 dots at CD19, that might suggest that, hey, this person, maybe they have a whole bunch of T cells, but when I look here, I don't really see anything in the area around CD19, right? That might be suggestive of potentially a B cell deficiency, right? Or if it was vice versa, if I had a whole bunch of dots at the CD19 mark, but if I didn't really have any density of CD3, right, then I would be more concerned about a T cell deficiency, right? So that's kind of how you read these. Again, these are just markers to kind of tell you what cells you're dealing with. Remember, CD3 is for T cells, CD19 for B cells. Now, these B cells, not only will they have a B cell receptor on their surface, which we already said is going to be membrane-bound IgM, or in some cases it can be IgG, and we'll talk more about this in the next video. But you can also have the MHC2 receptor. Remember, the MHC2 receptor, the whole point of an MHC2 receptor is so that you can present antigens to T cells primarily. Remember, B cells are also antigen-presenting cells. It's not just dendritic cells and macrophages, right? B cells can also present antigens. So like, let's just say, for example, how would that happen, right? If I had some antigen floating around in the body, right? Here's some antigen that then binds to this antigen binding site on this antibody, right? So it would bind to this variable region. So this is going to be, the perfect match for this immunoglobulin B cell receptor. So then that antigen can get internalized into this B cell. And then what that B cell can do is it can take its MHC2 receptor. Okay, this is my MHC2. Okay, so this B cell also has these MHC2 receptors, so it can internalize that antigen, and it can take that antigen, right, and then it can present it on the MHC2, and then it can take this antigen to a T cell. So once these B cells bind to this T cell, for example, right, then we'd get a proliferation signal, and they would go to the lymphoid follicles, and we talked about this. Primarily go to areas that are very active, secondary follicles. You'd have germinal centers that are forming, where you'd have a lot of B cells that would be undergoing affinity maturation, isotype switching, and I'll talk about that also in the next video. And finally, activated B cells can become plasma cells. Even activated B cells that don't involve T cell interaction can do this, and they can produce and secrete antibodies, which again, I'll also talk about in the next video. But I think this at least gives you a high-level overview of what we're talking about in terms of B cells. And like I said, next video we'll go into more of the details. Here are the references for this video, and we'll see you in the next one.